critical species, reactive species, that kills the plant. Rest O2 is thrown out of the
Right.
liquid, right?
Right.
Now that O2 once enters through an air compressor or a blower, it can facilitate the aerobic process.
Yeah, right.
That's a built-in contradiction of principles.
Exactly.
Yeah. There is one more ambiguity I find with engineers with reference to BOD. And I don't know whether you are or you must be conversant perhaps with sewage treatment. Aeration part is 70% of the treatment cost. And in my view, aeration has no whatever. Because aeration will help the air breathers like paramecia, MEP,
Yeah, it will further encourage their growth and boost them up.
means, it means eukaryotes.
Right.
Prokaryotes are not air breathers. In aerobic prokaryotes, they use the oxygen which is bound to another element like CO2, yeah, nitrate, which is bound to nitrogen, bicarbonate, SO2, SO4, NO3, NO2, etc. Pure O2 is an unbound molecule. So when I make a statement that environment or atmosphere has no oxygen, people go mad at me. But
I can see why. Yeah.
O2 is a molecular oxygen. It's not an elemental oxygen.
Mm-hmm.
Now, I was going deeper and deeper into this. So then I ended up with aerobic prokaryotes. Because eukaryotes have no role in BOD reduction. They will grow. And they will grow means they will add to the molecular mass of the entire liquid volume. So it's not degradation, it's a building up process. That is why we call it pranvayu. So oxygen helps to build, not to break or
Right.
destroy. So the first aerobic system that appeared on the planet was CO2 plus H. Later become CO2 plus H2O, that was photosynthesis. But photosynthesis is also viewed at in a very restricted sense. Because we take photosynthesis in general sense as oxygen giving or glucose forming. That's not true. I have my retinal cells, rods and cones in our eyes, right? They react to photons.
Exactly.
And
Yeah.
they send an electrical message to the brain that there is something. If it's a flower, it is red or pink or purple or white, whatever. So, photosynthesis in its limited sense, viewed as oxygen performance or production factory, it was a flawed idea. Earlier prokaryotic photon response was neither oxygenic nor glucogenic. There were basically C2, C3 molecules, pyruvate and right. So the early photosynthesis is reverse Krebs.
Okay.
The Krebs cycle that we were taught, glucose to pyruvate to pyruvate, etc. And then now that clockwise is a later appearance. Because glucose is broken down to CO2 and H2O. Now, where was glucose? There was no glucose.
No glucose in the
So, the
food. Yeah.
building part, the first step of C1 to C2, C3 molecule for construction, it's a building process. So, aerobic is the one which utilize the reactions or microbes which utilize oxygen as a terminal electron acceptor, in an electron transfer chain. Now going back to, then I raised the point in your session that all the eukaryotes are useless as far as the breakdown of organic molecule is concerned. Because glucose is broken down, but ATP is released, and ATP is utilized to promote higher functions of the body.
Yeah.
So it is a part of the breakdown, building process. Now, if we see the labeled carbon C14 of glucose, that enters this process reaction. In a breakdown, CO2 is also C14. It's a labeled carbon. But in our biochemistry that we study through acetyl coenzyme and all that, that carbon after pyruvate to PEP, phosphoenol pyruvate, it goes to amino acids, carbohydrates, lipase, and that carbon is labeled. CO2 that emerges is normal carbon.
I can see, yeah.
So the aerobic process and labeled carbon path is more towards the building, not CO2. Or methane. Even methane, see, carbon is, it will be having labeled carbon only if it follows the breakdown. And now breakdown is anaerobic.
Breakdown is anaerobic, right.
So air has no role. It's all about the electron shift from either somebody gains it or someone loses it.
Right.
And that is governed by the redox potential of the whatever molecules involved. Coming to our point, that the breakdown was the sole domain of the prokaryotes. Prokaryotes are not air breathers, though they are aerobic. But that aerobic have nothing to do, because oxygen appeared on the planet 2.33 billion years ago.
Yeah, much later, yeah.
So what was that before? Or there are no aerobes? Or there were aerobes? What was the source of oxygen? Now we came to this symbiont theory. This bacterium is engulfed by archaea and all that. So this bacteria, which we call it mitochondrion today, it was a prokaryote once upon a time, and therefore it was non-oxygen breather.
Right.
Or we define that it was not a bacterium, it was air breather. Then how it appeared on the planet? We'll have to see the lineage of the whole. Then after we discussed, I asked question and I was reading. Now, prokaryote ribosomes 16S, they are cell attached to cell membrane. And 18S ribosomes are attached to endoplasmic reticulum. To C18S, something to do with the membrane evolution in its molecular architecture.
Okay, yeah.
And I'm unable to get the link because I'm reading in a room sitting here and just try to question. And Dr. Snell is kind enough to talk very frequently with me, and I enjoy it. I really thank her because she's exposing me to many other scholars in the. But whatever IITians and engineering people that I have come across, they are mainly mathematicians. So they work more with calculator.
Right.
And microbial system requires lot of reading. If you sit with a calculator of highest order, no photosynthesis or Krebs cycle or glycol. And glycolysis and fermentation appeared later in the evolution. Carbon dioxide, single carbon, to pyruvate, which is a 3-carbon molecule. For first 2 billion years, it ended there and then came down. It was only when this blue-green algae appeared, then C3 became C6.
C6, yeah.
C6 is a later part of the evolution of carbon story. And it is once C6 formed, then glycolysis and fermentation.
Right.
So fermentation is later in the origin of molecular symphony.
Exactly.
Now it's that story. If we see, I'm relating it with the history of the planet and geological. And all that origin of life appeared at the bottom of the sea. And there was no environmental O2, and therefore there was no dissolved oxygen. And therefore there was nothing like oxygen breathers. Either aquatic or for protozoans came hardly 700 million years ago. On the contrary, jellyfish came 850 million years ago. And sponges hold the secret because they have cells with 16S and 18S.
Okay.
So the sponges first came, highly specialized group, which on one hand gave rise to protozoa. Because one more point, information that supported my objection is, no prokaryote has a phagocytic membrane. They are fixed shape, not changing. So, a phagocytic membrane can engulf something. That process is not clear.
Yeah. Right.
Because
I can see you. I can understand. Yeah, what you're trying to say. Yeah. Okay.
the stiff membrane cannot engulf anything.
Exactly.
It
Yeah.
can eat something and break down, yes. But then there is no symbiosis if it breaks down.
Exactly. So then the endosymbiotic theory, we don't know how
It goes for a toss.
Yeah. I never thought it this way, sir. So thank you for pointing this out to me. And Sneha ma'am, welcome. Good evening.
Yes, good evening. I'm looking forward to the question which Ravalji is always raising about 16S to 18S. So I want to hear from you what was the work that you had done and how do you would like to answer to this question of Ravalji.
Yes, so the thing is, well, this is such a novel line of thought. Honestly speaking, I never thought in this line. Like I have worked like in RNA related topics. Like I have worked on how ribosome facilitates protein folding, bacteria. I had worked on bacteria mainly. So I'll show you part of that work also. That was part of my PhD thesis work and the work I was doing in US in my postdoc and after I came back also. But right now I work in a rural college where I don't really have much research facility. So I work on a completely different field now.
So
Like I
you,
work
you,
with
you can
sugar, wood,
show
honey.
your work that you had done
Yeah,
and
so
further we
I'll
can.
show you both the PowerPoints. So the one I presented actually, let me share my language presentation sessions actually. So my slides are, can you see my slides?
Yes, we can.
Okay, let me take it to slideshow mode. Visible?
Yes, good.
Yeah. Okay, so sir has already seen this, but I'll just go through the slides quickly. So basically, this work it deals with EIF6, which is generally thought to be a translation initiation factor. How that actually is acting as an essential 60S ribosome biogenesis factor, and it links calcium signaling with this ribosome biogenesis process. So This EF6 is highly conserved, so my slides are both in English and Bengali because I presented in the regional language sessions. So EF6 is highly conserved from yeast to mammals, and when it was initially isolated and purified, it was on the basis of its property that it binds specifically to 60S subunit, blocking its association with 50S. So, based on this anti-association function, it was thought that it plays a role directly in translation initiation, to providing a pool of free subunits during that initiation phase. But when with a deeper probe into the cellular role, it was found that it does not act as a canonical translation insertion factor like EI5 or EI3 or others. Its role is actually elsewhere. But this is how it was. This is the property based on which it was initially isolated. That it acts as a 60S, 40S anti-association factor. It binds to the same region where 40S binds on 60S, thus blocking the association of 40 to 60. So when the human cDNA and the Saccharomyces cerevisiae yeast single copy essential gene, which is called TIP6, they are both cloned, and they are 72% identical in their amino acid content, and they have 245 amino acids each, and based on this similarity and conservative nature, molecular genetic study was done on yeast to further elucidate how it functions. And then it was found that when a conditional null allele was made, because it's a single copy essential gene, that even if we deplete ESTF6, then the yeast cell growth is inhibited and the rate of in vivo protein synthesis is being affected. But it still can translate proteins in vitro. So it was not directly inhibiting translation initiation, then protein synthesis would have stopped completely. So this is basically the ribosome biogenesis pathway. You all know it. So this is for the students mainly, so I will skip this slide. It was found that in the nucleolus, while the rRNA is being synthesized, if EF6 is not present, then this processing of 27S to 5.8S and 25S, this is inhibited. And these are both constituents of 60S. So 60S biogenesis is hindered. So if you do a polysome profile, what happens is, in the absence of EF6, 40S remains unaffected. Its synthesis is unaffected, but 60S biogenesis goes down, so does 80S, and instead of polysomes, what appears are half-mar polysomes, which are basically the 43S initiation complex waiting for the 60S counterpart. But because there is a deficit of 60S, that never happens. And so this is the polysome analysis. So if we deplete it of a canonical translation insertion factor like EI5, then polysome also goes down, and so does 60S and 80S both. But here what we see is that 60S is selectively going down with the concurrent appearance of half Mars. And then on repair analysis, it was like with radioactive labeling and all that part was elucidated, that this is the step which is getting hindered. Now, not only that EF6 is essential for this 60S rRNA processing, that is in a ribosome biogenesis, which is basically a nucleolar function here, it is also essential to escort the nascent free 60S from the nucleus to the cytoplasm for final maturation. So it has to continuously shuttle in between nucleus and cytoplasm. So you know that when ribosome biogenesis is going on in the nucleus, a number of transacting factors are also involved who associate and dissociate in a very ordered sequence. And when pre-60S is coming out from the nucleus to the cytoplasm, then a very few transacting factors still remain associated, and this EF6 in yeast it is TIP6B, and the nuclear export adapter NMD3 are the last two proteins which are still associated. and they are the last which get dissociated in the cytoplasm. Thereby the pre-60S now becomes mature translation competent 60S and can take part in translation, and then the EF6 has to be imported back to the nucleus for continued 60S biogenesis. So EF6 has to undergo well-regulated efficient nucleocytoplasmic shuttling. Two other proteins are also involved in this process, namely in yeast, EFL1P and SDO1P. Now this SDO1P, its counterpart in mammals is the mammalian SBDS protein, whose mutation results in a ribosomopathy, where 60S biogenesis and maturation goes down. It's called Schwackman-Bodian-Diamond syndrome. Now in yeast it was found that these two proteins facilitate the release of the yeast EF6 from the pre-60S particles in the cytoplasm via structural rearrangement. Now after that, EF6 has to go back to the nucleus, but EF6 does not contain any NLS or NES. But other proteins have been shown who do not have NLS or NES but yet undergo this nucleocytoplasmic shuttling, and that is regulated by phosphorylation dephosphorylation. Now similarly, EF6 is also phosphorylated at two very conserved serine residues at positions 174, 175, and that phosphorylation takes place in the nucleus. It is done by the nuclear casein kinase 1 in yeast and also in mammals. And this phosphorylation is also essential for yeast cell growth and viability and for the nuclear export of pre-60S bound to EF6 from the nucleus to the cytoplasm. So on phosphorylation, EF6 comes out from the nucleus to the cytoplasm, escorting the pre-60S. Now, in the lower panel, you can see that the serine residues are highlighted. They are in bold. And very close to that, there is an LQVP motif in all nucleated species. And this is a potential binding site for a very well-known phosphatase, which is calcium calmodulin dependent calcineurin phosphatase. So I'll come back to that later. This is where I'm trying to show you that EF6 in mammalian cells, it is predominantly cytosolic. But if we inhibit its phosphorylation, either by mutating the serine residues to alanine, or we inhibit CK1 kinase, there is a kinase function by adding D4476, which is a specific inhibitor, then it gets stuck in the nucleus. So phosphorylation is essential for the nuclear export. So we wanted to see whether the reverse process, dephosphorylation, is done by calcineurin and whether that dephosphorylation promotes nuclear import. So, as I said, this putative binding motif, LQVP, for calcineurin phosphatase is present in the EF6 in all nucleated species. And the calcineurin phosphatase depends on calcium and calmodulin for its activation. So if we treat mammalian cells, we have tried a number of cell lines, and in every case we found that if we activate the cells with calcium or with calcium ionophore like ionomycin, then nuclear import is being facilitated. Now, calcineurin binds to this LQVP motif, which is actually LXVP except any amino acid. In EF6 it's LQ. Calcineurin binds to its target site on its target protein at a hydrophobic pocket formed at the interface of calcineurins subunit A and subunit B. And that hydrophobic pocket of calcineurin is targeted by immunosuppressant drug cyclosporin A. So cyclosporin A is used as a specific inhibitor of calcineurin mediated dephosphorylation reactions. And so when we used cyclosporin A, then we found, yes, even in presence of ionomycin, nuclear import is being inhibited. And This nucleocytoplasmic shuttling, which is mediated by phosphorylation dephosphorylation events, that also takes place in presence of cyclohexamide. So it is not that new protein is being synthesized and that is going into the nucleus. No, it's a dynamic process. And because it involves calcineurin, so it also involves calcium signaling in principle. But we are yet to show that yes, calcineurin is indeed dephosphorylating EF6 to facilitate its nuclear import and further participation in the 6-TS rRNA processing. So this is with HeLa cells. So if we do a Western blot analysis of HeLa cell extract, then we see that endogenous EF6 migrates as two immunoreactive bands. And if we add CIAP, calf intestinal alkaline phosphatase, then the upper band disappears, suggesting that a fraction of EF6 is phosphorylated in vivo. Similarly, if we incubate mammalian cells with calcium or calcium ionophore ionomycin, that also results in gradual disappearance of the upper band, slower migrating band, suggesting that activated calcineurin is dephosphorylating EF6. But if we add the specific inhibitor, cyclosporinate, to the system, then even in presence of ionomycin, calcineurin cannot dephosphorylate it any further. So here you can see that we have ionomycin. So till 30 minutes, some of the dephosphorylation has taken place, but then if we have added cyclosporinate, then it does not go down any further as it does here. So then we made a mutant, triple mutant, where we mutated the LQVP site to AQAA. And both the wild type EF6 and AQA mutants, they express in the same levels, and they are both cytosolic predominantly. But if we activate the cells with ionomycin, that is we activate calcineurin, then if we do an IP, then wild type EF6 can associate with calcineurin. Calcineurin is tagged with HA and EF6 is tagged with histidine. So you can see that wild type EF6, when in presence of ionomycin, that is calcineurin activation, it is associated. But because in the AQA mutant, the calcineurin binding site is mutated, we cannot see any association, and also the nuclear import does not take place. It remains cytosolic. So this further confirmed our belief that yes, activated calcineurin binds to EF6, dephosphorylates it, and then facilitates its nuclear import. And then we wanted to see whether this nuclear import also involves calcineurin. And when we did an IP of the nuclear extract, we found that activated calcineurin remains associated with EF6 even in the nucleus. So it is kind of piggybacking EF6 after dephosphorylating it, so that it can go back to the nucleus, because EF6 does not have any NLS. So it is kind of piggybacking, and probably it is staying there as a complex, antagonizing the effect of the nucleus CK1, because on phosphorylation EF6 will come out, but EF6 has to do its part in the RNA processing. So till EF6 is once again ready to be transported to the cytosol, presumably bound calcineurin is antagonizing the effect of nuclear CK1. Other factors may be involved. Still, we need to work on that to further work out the details. So this is my model, that in the nucleus there is a large precursor RNA which is processed for pre-40S and pre-60S. And during the processing of pre-60S, one of the essential transacting factors is EF6. And EF6 is also needed to escort the pre-60S from the nucleus to the cytoplasm, where two proteins, SDO1 and EFL1, facilitates its release from the pre-60S. And this function of this SDO1 in mammalian system, that is SPPS, leads to the ribosomopathy SPDS. And once AF6 is released, then calcium signaling takes place and gastric phosphatase comes in, dephosphorates it, and probably there may be other factors involved. It goes back to the nucleus together as a complex, where EF6 can continue to work in the pre-RNA processing state till it is ready to be phosphorylated by CK1 and come out as caught in the pre-60S. So this is the part I actually presented in that session.
Yeah.
So,
So I could see that very elaborated study you had done, but very specific to. But then tell me, what was the application that you had in mind while you designed this particular proposal and you worked on it?
okay, so first of all, I'm very interested in ribosome. Like from my PhD, I've been working with ribosomes, and these days more and more ribosomopathies are being reported, and these are rare autosomal diseases. But what I find is very surprising that a basic function is inhibited, but the symptoms are very tissue specific. I mean, skeletal muscle is affected. Sometimes it results in bone marrow failure, and it's that serious. But it's very specific. I mean, but it's such a generalized function. So that is one reason why I'm interested in ribosomopathies, and yes, it has been shown to be implicated in a number of ribosomopathies like this one. Another thing is that in many cancer cells, because they have to grow so fast, so they have to synthesize more and more proteins. So their ribosome synthesis goes up. So if we can regulate an essential ribosome biogenesis factor, because clearly these are all regulators, these phosphorylation, dephosphorylation mediators, they can regulate EF6 functioning. So by that, if you can do a targeted regulation of EF6 and thereby bring down 60S biogenesis synthesis and ribosome biogenesis in cancer cells, that can probably be provide a therapeutic platform. So, but clearly much more work needs to be done, and as I said, like right now, I don't have any option of working with
No, but I
mammalian
think
cell lines and systems.
you will be able to much more relate what Ravalji had posed the question, because I see when you talk about the dephosphorylation and phosphorylation aspects, and you are talking about the 40S to 60S and then EIF6 involvement. So in that case, I think if you relate and if you can little you may be having that kind of an idea that that 16S to 18S about that ribosomes transition. So I think you will be able to, through your studies, whatever you have deciphered, I think little bit if you can put light on that aspect, what it comes to your mind.
Yeah, so I was wondering if you can like give me any related study material, like if people have tried to work on that. Oh, let me show you another study. So it's visible.
Yes, yes.
Okay, so my PhD lab, they work on ribosome mediated protein folding. And what they have shown over years, and there are many publications from our lab, I can share with you if you're interested, that ribosomes from all sources, they can act as general protein folding modulators. Of course, there are specific protein folding modulators like the established ones, but ribosome plays a basic role, making the protein, the nascent proteins it's synthesizing, competent to go into that specific protein folding pathway before it becomes functional. So it was shown in bacteria. We worked with bacteria basically. Like I worked with bacteria. That the active site for such protein folding overlaps with the peptidyl transferase center in the domain 5 of E. coli 23's RNA. And during protein folding, I also showed that the unfolded protein helps to dissociate the 70S ribosome into 50S and 30S, and thereby it sequesters the 50S subunit to access the PTC to attain a folding competent state. And For efficient protein folding, this PTC then needs to be fully open and accessible to the folding protein. That is, it has to be free from other binding factors, including tRNA. I showed that if tRNA is still bound to P or E sites, then ribosome mediated protein folding is inhibited. So I'll show you a part of that. So just as an introduction, what our lab had shown is that the active site of protein folding is the domain 5 of 23S RNA on the 50S subunit. And if you try out different antibiotics, the 30S bound antibiotics do not inhibit the process, but the 50S bound antibiotics which bind in this domain 5, they inhibit also protein folding. So what we did was we split domain 5 into two parts. One which comprises mainly the central loop that overlaps the PTC and binds the, like we showed it with denatured protein, but you can think of it as nascent protein which is not yet folded. And that part we called RNA1, and the other part we called RNA2, which helps releasing the protein into the on pathway of folding now. And if this step is missed, then the protein goes on to misfold. We showed that with DNA, K1, etc. That's another story. And only a conserved set of nucleotides of domain 5 rRNA binds specifically to unfolded proteins. Now, during protein synthesis, tRNA molecules, they also interact with different bases on domain 5. And the main interactions between domain 5 and the peptidyl tRNA, that is in the P site of 50S, they are limited to a Watson-Crick base pair between G225 and C74. I'll show you. There's a non-canonical interaction of U2506 2585 with A76 of tRNA, and a contact between A2451 and the amino acid moiety of peptidyl tRNA. There are also some additional bases which also are protected in the process. So this is from one of our publications. The figure shows 70S, it's 50S and 30S subunits, and the peptidyl transferase center, the domain 5, and the way we have split it into two parts, RNA1 and RNA2. So you can see RNA1 is the one with the central part, loop and all. And this is the secondary structure of the PTC, and the 5 major sites which are involved in protein binding during protein folding, they are highlighted here. Now, what I did was, like this is one part of my thesis work, as I said. So this was to study the interaction of tRNA and unfolded protein with PTC. So I wanted to find out why tRNA is present, still bound to the PTC, can ribosome fold an unfolded protein. So how we did that? The strategy was that we unfold a protein and then we try its refolding by 70S ribosome. We tried it with deacylated tRNA bound to P and or E sites. So there's a protocol by Noller and his group, where if you use higher magnesium concentrations in a poly-U independent manner, you can this tRNA can bind to P and E sites. And then we extended the studies to 50S, to its 23S, to the domain 5, and finally to the RNA1 RNA2 species. So this was our experimental procedure. We took BCA and we unfolded it in 6 molar guanidine hydrochloride and confirmed that the secondary structure is lost by CD spectral analysis. And then we tried to refold it using ribosome or its rRNA, using mostly the same concentration as that of denatured BCA. Now, deacylated tRNA, in absence of any mRNA, can bind to E site of 70S at 20 millimolar magnesium, and at 25 millimolar magnesium it binds to P and E sites. These are like established protocols. And we can confirm the binding by cellulose nitrate filter binding assay. And then we assayed the activity of the refolded enzyme, which is been refolded by folding modulators with or without increasing concentrations of bound tRNA. We assayed that by adding 500 micromolar paranite to phenyl acetate to each set, and because we are using BCA, and then measured the increase in A400 nanometer with time. And then we calculated the extent of refolding the denatured protein by taking the ratio of the activity of the refolded enzyme to the activity of the same amount of native enzyme. And this is where I'm trying to show you that by filter binding assay we monitored that ribosome tRNA complex formation is taking place, that deacylated tRNA is binding to ribosome at high magnesium. So both 70S, 50S and all that we confirmed. And we found that tRNA when bound to wild type ribosome or 50S or the RNA component, they all inhibit protein folding. That while tRNA is bound to the PTC, then ribosome cannot fold the unfolded proteins. Same with the RNA1 and 2 species. And when if we mutate the 70S, then also we find that only when the ribosome is the wild type ribosome and tRNA is bound to it, then this inhibition takes place. Because in the mutant ribosomes, what we are doing is the tRNA binding sites are mutated. So even in the presence of high amount of tRNA, it's not binding and so it's not inhibiting the folding process. And for tRNA to bind to the wild type ribosome, its 3' CCA end is also necessary. If we don't have the 3' CCA end, or if we mutate the 3' UCA, 3' CCA to UCA and then add the mutant ribosome, thus neutralizing the effect of the mutation, then again we are getting inhibition. So all this we use to confirm that yes, when tRNA is bound to the PTC of ribosome, then its folding property, folding the unfolded protein, that is being hampered. Both the binding of the unfolded protein and the efficiency of releasing the bound protein, they are both affected. And this is very interesting that we found that when we are working with wild type 70S and we are trying to do the protein folding studies, then in the process 70S gets split into 50 and 30. And then the unfolded protein can access the free, open, accessible PTC part to get folded. So I also tried to find out that it is that whether the in the last step of protein synthesis, when all the parts are being dissociated, 70S is also dissociating into 50S, 30S, where clearly the unfolded protein which is trying to get it folded is helping in the splitting process. So I was also trying to work out that part, but then I got an offer from Albert Einstein College of Medicine for postdoc and I left. So others have carried out this part and they have also published that. So basically through this work what I attempted to find out is whether 70S ribosome can refold unfolded proteins while its domain 5 is occupied by tRNA. And my preliminary findings suggested that deacylated tRNA molecules bound to P and E sites of domain 5 inhibit protein folding by 70S and the RNA components, and also it inhibits the splitting process while tRNA is present. So I thought I'll share this with you also, because this also tells you the importance of ribosome not only in synthesis but also in the initial steps of protein folding, which is like all basic processes, and it kind of confirms the RNA world story very much. I'm very fond of RNA.
Yes, Munir Ravalji, please go ahead.
Can prokaryotes convert pyruvate to glucose? They can utilize glucose, break it down to pyruvate. That much I know. But the reversal of hexokinase function in prokaryotic cell, I have not come across a reference. So hexokinase has a reversible function with reference to eukaryotic yeast and other higher cells. I understand. And my major concern is sewage because each one of us produces 365 days. Nobody can say that I have no role in continuity. Antibiotic and all that is fine for the patients.
Right.
They are primed to handle greenhouse gases.
Methanotrophs.
CH4 to methylation and from methylation to many other signaling molecules and all that. Serine pathways and ribulose phosphate, etc. But the point is, if sewage treatment does not manage it, the greenhouse gases,
They will keep on piling. Yeah, they will keep on increasing. Yeah.
And world over, STPs have 8 times more greenhouse gas release than in nature. For example, in paddy or in mangroves. So, and one interesting mechanism I've learned about, it is the microbes in our colon that ultimately break down higher molecules to single carbon. And if denitrification is not properly done, then nitrous oxide accumulates in the colon and that ultimately gives an effect known as Alzheimer and all that. So, greenhouse gases, even besides climate instability, they can create a havoc. All neural synaptic junction functioning is interfered due to nitrous oxide not released properly or not converted to nitrogen. Because nitrogen is inert, so is O2. That is why they are more in the atmosphere. And CO2 is the driving gas towards origin of life. That is why we have CO2 to the level of 0.04%. So body, bacterial life, vegetation, animal life, whatever. What is useless to them, they throw it up. And that is nitrogen and O2 is an excretory product of chlorophyll. Now, the cellular respiration, even in plant cells, is C6H12O2 O6 plus O2 is equal to CO2H2. Now, if this equation is right and that O2 is required, why would plant cells throw away as excreta? So, C6H12O6 are glucose plus O2. That O2 needs to be understood in light of O2 which is thrown out into the atmosphere.
Whether they're the same and being recycled by plants and all.
So O2 is a bound oxygen. It's not unbound. Unbound O2, if you want to pull apart 2O, O2 separated into 2O, the energy required is 495 kilojoules. No cell has that energy.
Yeah.
To promote one reaction.
Right.
So I'm stuck with 16S only. Even 50, 30, 70 and 80S ribosome is a very, very higher architecture. 16S is a very adamant protein. That ribosome protein is so adamant that it has not undergone a change for billions of years.
Right.
And we still hold, even in our running, suppose an athlete is running, the somehow aerobic reactions are not properly supported, then pyruvate becomes lactic acid, and
Lactic. Yeah.
we have a muscle.
Cramps and all. Yeah.
So that legacy even is there with us, which are we are the latest creation in the evolution. So I will be very grateful if you can send some references, because I'm reading Pablo Rampelotto. His book is Microbial Evolution. I mean, Evolution of Microbial Genomics. That is the title. And he
Okay.
vehemently argues this symbiosis theory,
Okay.
endosymbiosis.
Endosymbiotic.
Yeah. So, and unless we understand this, because the entire difference lies with prokaryotic ribosomes and eukaryotic. All protozoa, worms, and all that, they are eukaryotes. And entire sewage treatment plant is based on the eukaryote. And therefore, it is a DO-based principle, in short.
Hmm.
Billions of dollars per day is dedicated to aeration in sewage treatment. I cannot see a bigger stupidity in any part of human civilization, history of human civilization. Secondly, all the pandemics are urban in origin, and they are from the sewage. No forest
Yeah.
gives you a pandemic.
Exactly.
So, and the world and UNICEF and all, they also talk about cities as engines of the economic growth. I think it is the biggest catastrophe. I don't see that as an engine, because I don't see future civilization having cities anywhere on this planet if we want to be sustainable.
Yeah.
So of course my journey, mental journey, goes way beyond from molecules to ultimately we are concerned with survival, whichever
Right.
molecule or pathway. 20% of the
If you
aeration
understand
budget is
the
net
role of
saving.
16S.
Economically, we become rich. You see where I'm driving at?
Yes, I was wondering, ma'am probably knows about the. We have in Calcutta, like the East Calcutta wetlands, and
Yes, yes,
which is called the kidney of our city, and
exactly.
and it's mostly the bacteria which is doing the part, right? It's like you don't need anything artificial there. They are doing the job. So yeah. So
People use the word biotechnology. In sewage, what is non-biotechnology? Just tell me.
mm, exactly.
We can't. I can't put a screwdriver which will be a substitute for microbes. Ultimately,
Exactly.
only microbes will do the job.
Only my yeah.
Yes.
They are the tools, yeah.
The typical microbiology knowledge, which comes from the petri dish, means under controlled conditions.
Yes. Yeah.
Right? There is nothing controlled in nature. It's all wild. The nature does not know the term sterilized environment.
Right.
Sterilized environment is our laboratory condition. So I have, and I keep on discussing with Dr. Snehalji. She is a patient, the only patient listener who is willing to help me by reaching out to various experts in the field. But we are still fumbling as a species. If
You
you
know,
see
sir, like you remind me so much of my PhD mentor. Like when I was in the initial stage of my
it the
Equalize it.
best genetic tool to
Equalize.
understand genetic engineering. Because in one day you have so many generations. And E. coli does not have much of role in sewage management. It's an ideal genetic tool, not for sewage management. And not all E. coli are pathogenic. There is another fallacy that sewage engineers are not seeing.
yeah.
There are more than 700 serotypes, and very few are ideal host to become a pathogen. They are hosts to are susceptible to a viral attack. E. coli is not a pathogen. It is an ideal host
Yes.
which can mutate and become a pathogen.
Yeah. Yeah. Hmm.
So, I mean, in billions of liters world over, wastewater volume generation is 350 to 430 cubic kilometers.
My God. Yeah.
And you see, I have one wonderful book on cities where they have Michael and Smith, where they are the authors. They have mentioned Calcutta. What was the term, Snellji?
Pukur. That
You
was
would say
Pukur,
for
Pukur, Pukur.
Pupur, yes. And the most sustainable city once upon a time, it is Calcutta. So why we deviated from there and anyway. But I will need some experts like you in microbiology who only can help. I don't have any hope from engineers because they are talking with gears. If instead of 50 horsepower, they will talk about 500 horsepower pumping. But the
No, you know
company,
what the problem is, sir? Like the younger generations, I find they're like they're basically fund motivated. Whatever.
and that was his statement in 2018. So 2030 he put it as the last line, and we are still busy about making structures that we want to see, not what the
Martin
nature
Holliff
wants to
replaced Stephen
tolerate.
Hawking.
Anyway,
With all the knowledge and experience of yours with this study, the path you have tried to show us today, at least shown me today. Uh,
I am a beginner. For example, we all know that CO2, carbon in the center and two bonds on the either side to one O.
mhm,
So four valency of CO2 is satisfied.
hmm,
Now what happens to carbon monoxide? Is that carbon bivalent?
right.
Or if it is tetravalent carbon, then it should be a radical.
Exactly.
Plus
Some
2.
yeah.
I'm talking 8th standard basic chemistry.
Mm.
Just, and I have raised this question and people are fumbling. But fumbling, I'm not asking you anything.
You're making us think now, sir, in new ways.
Yes.
Like so many things, because we have learned it from our childhood, we take them for granted. We don't question them. But yeah,
No, so
I mean, this today was a very enriching evening for me.
questioning that will lead us to the next level of understanding.
Exactly. And as
What
long
I
as we
know,
have
I know. What you know,
gaps in our fundamental understanding, we cannot expect to understand the higher things better. No, we
then,
have to
then,
understand the fundamentals
then
fully.
academia
Yeah.
will have to accept this challenge.
Yeah.
Or we leave it to tech. Accept this challenge. Engineers are not equipped.
Yeah, I
So
can see your point. Yeah.
maybe we can make a group and stay connected through mails and all that.
Sure.
But I tell you, I am talking this for 30 years.
Hmm.
For the first time, I found you someone talking about ribosomes, and it to the level of a fraction of ribosome, not entire ribosome.
Hmm.
So I have very high hopes because I know it for sure that the ribosome 16S ribosome RNA rRNA is the answer. Engineers do not know what is a ribosome. They do not know what is a membrane also.
Exactly. Yeah.
So